All right, so that recording notification should be popping up. All right, so hi everyone, welcome to our last webinar in this set for the hippocampal subfields group. I'm really excited that so many of you have stuck with us through this, and for those watching later that you have come to the YouTube channel. Today, we're really excited to be featuring two amazing scientists, Dr. Maureen Ritchey and Xenia Grande, and we're going to have them share their work that focuses on the theme of functional connectivity. And Raban will give more of a detailed introduction in just a bit, but we're going to actually kick off by having a few announcements from the steering committee, which Dr. Anna Daugherty is a part of. So Anna, I will hand over to you for the time being so that you can share those updates.
Great, thank you, Kelsey. And if I haven't had a chance to meet everyone, let me just take a moment to introduce myself. And I'm Anna Daugherty, and it's my distinct privilege to currently chair the steering committee of this group. I just wanted to take a few moments to share my sincere thanks to our very large hippocampal subfield group, where we are more than 250 researchers strong, representing over 15 countries internationally. And this webinar has featured the connectivity of that group internationally over this last year. And so thank you so much for spending your time attending these webinars. I wanted to share just a few announcements about the group for things to look forward to. Over the next few months, we will be sharing different announcements and updates that include things about the protocol development, which is the main purpose of this group, and inviting opportunities for feedback and evaluation about some of those essential subfield boundary segmentation rules on MRI. We also will be resuming some of our working groups on different topics and adding new topics. And those will be online, as well as some possible in-person meetings as we all start to resume. These announcements will be made through our website and our listserv, so make sure you check back frequently. And if you haven't signed up already for our listserv, please check out the website for those links. This webinar series was a huge success in our view, because we got to see so much fantastic work and also have an opportunity to connect with new community members in our research environments that we otherwise didn't usually get to see at these conferences. And so inspired by that, we would like to continue this work after a brief hiatus. So this is our last meeting of 2021, and we are thinking about continuing this, resuming again in 2022. And to help frame that up, you will be receiving a questionnaire sent out from Kelsey and Robin, asking for your feedback about what you liked and possible topics. There will also be an opportunity to share your name if you would like to be a featured speaker on this webinar series in the future. And so also look for a call for speakers in the fall, in addition to that questionnaire, and we'll be trying to organize that schedule early on, and then resuming the webinar series possibly January, February of 2022. So with that and our small updates, let me wrap this up by saying a huge thank you to all of our speakers in the 2021 season. It was a fantastic lineup and just really phenomenal work that was featured. And extra huge thanks to Kelsey Canada and Robin de Flores. We as a steering committee had this idea for a long time, but it was only under the leadership and talents of Kelsey and Robin that it really came to fruition. And this has surpassed everything that we could have imagined. So all credit goes to Kelsey and Robin for your leadership, and just a tremendous amount of thanks. And so if you could join me in your reactions or some on-camera hand waves, thank you so much, Kelsey and Robin. And with that, I'll pass it back to you to introduce our speakers.
All right, thank you very much, Anna. So it's my great pleasure to introduce our first speaker for today, who is Dr. Maureen Ritchey. So Dr. Ritchey is an associate professor in the Department of Psychology and Neuroscience at Boston College. Her research is focused on the psychology and neuroscience of human memory. So she combines cognitive neuroscience methods, including fMRI and EEG, to investigate the relationship between brain networks and memory processes. So the goals of this research program is to determine how memories are organized in the human brain, to explain why we remember some things and not others, and to identify ways to intervene on memory-guided behavior. So today she will be sharing her work, putting the hippocampus into context, functional networks supporting episodic memory. So Maureen, the floor is yours.
Great, thank you for having me here. Let me go ahead and get my screen up. Okay, I think we're all good. All right, so today I am going to be telling you about some work we've done to use functional connectivity to shed light on the organization of memory systems. In general, we've been interested in how the hippocampus interacts with large-scale cortical networks to support different aspects of episodic encoding and retrieval. And since this is the hippocampal subfields group, I'll of course start with the hippocampus, and then I'll try to put it into context in terms of the connections with the rest of the brain. So as I'm sure all of you know, if you are in this webinar, the hippocampus has long been appreciated as playing a specific role in relational processing. So being able to form flexible associations between two things, like remembering not only that you've seen something before, but also where you've seen it and other associations that you may have formed at the time. But if we start to look beyond the hippocampus, which is quite easy to do with tools like fMRI, we can look at the medial temporal lobe cortex, for instance, which seems to be specialized for representing different kinds of content in memory. And namely, it appears that differences in inputs to the medial temporal lobes lead to differences in the involvement of cortical MTL regions in memory. So whereas the perirhinal cortex, which is at the anterior end of the parahippocampal gyrus, has been described as specialized for processing information about items, which largely extends from its connections to the ventral visual stream, the parahippocampal cortex, which is at the posterior end of the parahippocampal gyrus, has been described as specialized for processing information about context, and especially spatial contexts or scenes. Now, not only are these areas functionally heterogeneous, which we know both from rodent literature as well as analogous literature in humans, they're also heterogeneous in how they interconnect with the rest of the brain. So for instance, if you look at the functional connectivity of these areas, which is simply how their activity co-varies with activity in other parts of the brain, like the hippocampus or more broadly throughout, they seem to be connected with distinct corticohippocampal networks. So for instance, if you compare perirhinal and parahippocampal cortical connectivity with the hippocampus, you see that the perirhinal cortex tends to be more preferentially connected with the anterior parts of the hippocampus, whereas the parahippocampal cortex seems to be preferentially connected with more posterior parts of the hippocampus. And this is coming from data from Laura Libby and Charan Ranganath's lab. If you look at the whole brain, you see also dissociations in terms of the way that the PRC and the PHC are interconnected with different parts of the brain. So whereas the perirhinal cortex shown here seems to be strongly connected with ventral temporal areas, the parahippocampal cortex links up with areas more broadly through the medial and lateral parietal cortex, as well as in the medial prefrontal cortex. So we have described these patterns of findings as reflecting two corticohippocampal networks that are involved in memory. An anterior temporal network, which includes the perirhinal cortex, and as well as the amygdala and the lateral orbitofrontal cortex and ventral temporal polar cortex, that seems to be important for processing information about items as well as their attributes, including their emotional significance. And we can contrast this against the posterior medial network, which I've shown here in blue, which includes the parahippocampal cortex, the retrosplenial cortex, as well as other areas in the medial and lateral parietal cortex, that it seems to integrate the contextual relationships that make up an event. The hippocampus then is involved in binding item information supported by this anterior temporal network to the contextual frameworks supported by the PM system, and vice versa. So it seems to be playing this mediating role between these two cortical systems. So a key feature of this proposal is that we believe that these sets of regions are functionally integrated systems. that they play together in order to support a broad range of cognitive processes, including but not limited to memory processes. The posterior medial network in particular comes up again and again in neuroimaging studies. So it overlaps with portions of the default network. It pops up when people are imagining the future or other types of alternate scenarios. And it's super reliably engaged during episodic memory retrieval. So much so that some, including Nick Rugg, as well as other people, have called it the core recollection network. So today I'm going to tell you about some work we've done to characterize these networks and their relationship to episodic memory processes. I will start by telling you about a study where we used data-driven network analyses to see if our impression from the literature was right, namely to see if we could identify in a data-driven manner dissociable corticohippocampal networks, both at the level of large-scale cortical networks, but then also looking to see if we can use those tools to identify subregions within the medial temporal lobes themselves. I'll then talk about how we can link these networks to functional specialization during memory tasks. And finally, I'll wrap up by showing you evidence that these corticohippocampal networks reconfigure themselves during recollection as we bind specific details of an event into a contextual framework and are able to recollect that information. So starting out, I just want to give you a brief introduction to some of the principles of network science that are relevant here. So within the network science literature, there's an idea that within a larger network of connections, such as a social network, or here, the characters of Love Actually, there are smaller networks like friend groups or people who were in the same scenes of the movie. In these subnetworks, there are stronger connections within a network than there are between the subnetworks. So in network science, identifying these groupings has been referred to as community detection. And algorithms have been developed to apply grouping labels that maximize these within network connections while minimizing between network connections. So these algorithms have been applied to networks of people, but then they've also been applied to the brain, resulting in maps like this one of the cortical network organization across the entire brain. So we can also apply these methods to more narrowly defined networks. So you can zero in on a few regions that you care about the most and try to map out the subnetworks that connect those regions. And since we were really interested in how the medial temporal lobes were connected with the rest of the brain, that's the approach that we took. So in this study, we took a set of regions that we knew were functionally connected with the perirhinal and perihippocampal cortex. And we asked a simple question. If we apply community detection to the resting state connections among this set of regions shown here, which is an entirely data-driven approach, will we get back networks that look like our hypothesized posterior medial and anterior temporal systems? So to do this analysis, we first measured resting state functional connectivity between each one of these regions. So just looking at the correlation of the time series of these regions over the course of the resting state scan. And then we filled out the entire matrix of possible combinations between the regions, resulting in this functional connectivity matrix. So in this matrix, the warmer colors denote stronger functional connectivity between two regions, and cooler colors reflect weaker functional connectivity. So in this matrix here, I've already sorted it out by which regions the community detection algorithm said should go together, so that you can see that within a box here, there are brighter or warmer colors, meaning stronger connectivity, than there are between these boxes. So here I've color coded the regions according to the way that they were assigned by the community detection algorithm. So in this data set, we got back a blue network that looks relatively similar to the posterior medial system, so including these medial parietal regions, as well as regions in the perihippocampal cortex. We got this red network that looked relatively similar to our anterior temporal network, including areas in the anterior parts of the temporal lobes, as well as in the orbitofrontal cortex. And then finally, we got this green network that appears to be a ventral frontoparietal network that I'm going to set aside for the purposes of this talk. So more importantly, anyone can define resting state networks, right? But one of the things that we want to know as cognitive neuroscientists is, what is the function of these networks? Is there any functional significance of this organization? So in this study, we wanted to know also whether these networks played related roles in a memory task. So we took these same participants and scanned them while they completed an associative memory encoding task. And in this task, we simply asked them to encode associations between items and their features, their situations, or their spatial contexts. So then we measured encoding activity across the entire, I should have mentioned, then we tested their memory for these associations after the scan so that we could bin these trials according to whether the associations were remembered or forgotten. So then we can measure the encoding activity across the full set of 6 task conditions, which is 3 different types of associations and times remembered versus forgotten. And we can look at the pattern of brain activity that you see for any given region. So this is the equivalent of doing a region of interest analysis on our task data, where we just look to see in which conditions is this region most active. And for example, this region, the parahippocampal cortex, is most active when people were encoding these spatial context associations, and even more so when they subsequently remembered those spatial context associations. Now, importantly, we can do this for all of the regions in our analysis, just like any standard task fMRI study where you might have a lot of ROIs. But then the twist is we took these activation profiles and we treated them as vectors. And then we calculated how similar these activation profiles were for regions that were contained within the same subnetwork versus regions that were in different networks. So note that here, I'm not talking about task connectivity per se, but rather a simpler idea, which is just how similar would the ROI analysis results be between these different regions. So what we found was that within a resting state network, regions had more similar activation profiles compared to regions that were assigned to different resting state networks. So this implies that the similarity of 2 regions' functions during this memory encoding task was predicted by how connected they were at rest, indicating that in this case, the resting state was carrying information that was relevant to these regions' roles in memory processes. So we next wanted to know whether we could use a similar approach to define functional units within the medial temporal lobes. So this was a project that was led by Xiaofeng Wang, who at the time was a research assistant in Charan Ranganath's lab. And for this analysis, she took a slightly different approach to the data. So instead of trying to group regions of interest across the brain, she focused on slices, individual slices along the long axis of the parahippocampal gyrus. And then she used a slightly different algorithm to group these slices, which I am going to skip over the details of in the interest of time. But more or less, she calculated the pattern of voxel-wise connectivity with the rest of the brain for each individual slice, and then compared these patterns between the slices. So what she ended up getting from this analysis was that if you do this type of comparison and then you try to look at which slices end up lumping together in terms of showing similar profiles of functional connectivity with the rest of the brain, then what we see is that there were 3 functional units within the parahippocampal gyrus. One that mapped onto anatomically defined segmentations of the parahippocampal cortex, and 2 that seemed to define to anatomical definitions of the perirhinal cortex, one that was more anterior and one that was more posterior. Now, importantly, these 3 chunks also play different roles in memory. So just to remind you, we had also scanned these people in this memory encoding task. This is the exact same data set as what I had shown you before. And what we ended up seeing in this analysis is that whereas the most posterior chunk here, corresponding to the PHC, predicted subsequent memory for spatial associations, the most anterior chunk supported memory for item feature associations, consistent with a hypothesized association between contextual processing in the posterior part of the parahippocampal gyrus and item processing in the more anterior part of the parahippocampal gyrus. So note that these subdivisions were defined entirely through this data-driven analysis of the resting state functional connectivity data, and yet they were informative with respect to regional specialization in our memory task. Now, I should also note that when I was initially looking at these data, I didn't see this item feature memory effect come out. And that was partially because I was looking at the whole PRC together. So this is really something that became apparent only once we used the functional connectivity data to refine our region of interest. So this study and the one before it, I think, provide a good starting point for how to define functional networks and think about their implications for memory. But more recently, Alex Barnett and Charan Ranganath have extended this approach even further, identifying 4 subnetworks that were all functionally interconnected with the hippocampus, albeit with different patterns of connections across the long axis, suggesting that this original anterior temporal versus posterior medial distinction may have some variability within that as well. So there may be actually subnetworks within these 2 larger scale networks, so we can refine our understanding of how these networks are organized. And in my lab, similarly, Rose Cooper, a postdoc who was in my lab, also identified subnetworks of the posterior medial network that played dissociable roles in event perception. So I think considering the subnetwork organization of these larger scale systems will be a useful path forward for generating new predictions about the functional significance of these cortico-hippocampal interactions. Okay. So turning back to this framework I outlined earlier, one key prediction is that both sides of these networks, so both the anterior temporal side and the posterior medial side, are important for reconstructing the details of an episodic memory. You need access to information about the specific entities associated with the memory, which should be carried by the anterior temporal network, but you also need to know how to piece them back together into a coherent event. So in other words, these networks need to be able to interact with one another. And this is where the hippocampus is thought to come in. So just as the hippocampus serves to bind features into episodic memory at coding, it's also thought to relay information to and from these 2 networks in the service of recollection. So in this next study, which will be the last study that I talk about today, we set out to test these very ideas, that these networks need to interact with each other and with the hippocampus to support this kind of multi-dimensional memory retrieval. So to test this idea, my postdoc, Dr. Rose Cooper, developed a paradigm to test memory for complex multimodal events. So in this paradigm, we wanted to be able to assess memory for different kinds of details, including item information, spatial information, and emotional associations. And we also wanted to be able to assess memory in a fine-grained, objective way so that we could really ascertain the quality of memory above and beyond a simple yes-no recognition test. So basically, we wanted it to feel like a real event, but we also wanted to have total experimental control, because I think we're a little bit of control freaks here. So we scanned 3 participants while they encoded a series of objects, each one associated with 3 features. So each object was associated with a color. So this is just the color that the object was presented in during encoding. So you can see this is a reddish color. This is a more turquoise-ish color. The object was also associated with a spatial context, which was defined as a particular viewpoint within a 360-degree panorama scene. And then finally, they were associated with an emotionally negative or neutral sound. So this was the encoding experience. And then shortly after encoding, we tested their memory for each object by first asking them to remember each event in as much detail and as vividly as possible. So we simply cued them with the object shown in grayscale and told them to remember everything that they possibly could. And then we followed this up with specific questions about the emotional context of the object, as well as questions asking the participants to reconstruct the item's specific color, as well as the spatial context in which it was presented during encoding. So these latter memory tests, I think, are a lot easier to see if I actually play you a video of what this looks like for the experiment for the participants. So just imagine that this is the event that you're encoding. So you see this kind of turquoise-ish radio superimposed on this living room scene, and you're hearing a negative sound. And then during retrieval, before we start this retrieval task, I want you to just try to mentally reconstruct what color was the radio, picture the scene as well as you can. And this is what that looks like for the participant. So they first were given that remember cue. Then they were tested on their memory for the emotional context. And then finally, they could dynamically reconstruct the color of the object by using the buttons in the scanner. And they could also reconstruct the visual angle that they had seen within the panorama associated with this object. So this was a really dynamic, engaging task in that people were able to actually control their ability to reconstruct the image that they had studied as closely as possible before. Okay. So then to analyze the behavioral data from this study, what we wanted to look at were people's errors in making their responses for the item color as well as the spatial context. So if the item had been presented in this magenta color and they gave a response showing that it was orange, this would be some degree of errors apart. So this is about, let's say, 70 degrees or so. Similarly, with the spatial context, we can measure their error in replacing the object within that 360-degree panorama. So this gives us really fine-grained objective data about how similar their memory is, or at least what they're reconstructing as their memory, to what they had studied before. So the first thing that we wanted to test in this study was whether or not the functional network interactions were going to be reorganized during episodic memory retrieval. So in other words, we wanted to know, is there going to be a change in the way that the anterior temporal and posterior medial networks interact to support this recollective process? So to do so, we ran a background connectivity analysis that let us look at the baseline network interactions, irrespective of what was going on for the individual trials during both encoding and retrieval. And in this analysis, we were focused on anatomical regions of interest in the posterior medial network, shown here in blue, the anterior temporal network, shown here in red, as well as in the hippocampus. So during encoding, we found evidence for network modularity. So regions within the posterior medial network were more connected with each other than to regions within the anterior temporal network, and vice versa. So this pattern of results looked familiar because it echoed the results that I had shown you before that were based on the resting state data. So during encoding, it appears that we see the same type of network modularity and that the PM and the AT systems pull apart from one another. But then critically, we wanted to know what's going on during memory retrieval. So when we looked at retrieval and then compared retrieval directly to encoding, we found that modularity went down. So in other words, during retrieval, the two networks became more correlated with one another, suggesting that retrieval involves integration of the PM and the AT networks. And in addition, these networks were also more strongly connected with the hippocampus during episodic retrieval. So this was a background connectivity measure, which specifically removes tasks or effects related to the task design. So this particular analysis wasn't telling us anything about how these changes were associated with memory performance. So to do that, we next looked at how memory-related changes in functional connectivity using a psychophysiological interaction approach, or PPI. Now, importantly for this analysis, we focused on changes in functional connectivity during this remember period, when the participants were trying to remember everything they could about the event without any additional information being provided. And we calculated a composite measure of the quality of their memory, which included the number of the features that were recalled for each object, and also scaled by the precision with which it was recalled, precision being derived from that error measurement that I was telling you about before. So in line with some previous research, we did find that there were widespread increases in functional connectivity that were associated with memory performance, and specifically with our continuous measure of behavioral memory quality. And we saw that these increases were especially pronounced in terms of the way the PM and the AT networks interacted with the hippocampus, as well as how the PM and the AT networks interacted with each other, rather than changes within the networks. So on the left, I have this broken down by network. Then on the right, I'm showing you connections between the individual regions. So you can see that the strongest changes associated with memory quality were mostly between the hippocampus and regions in the posterior medial network, as well as region connections between the AT and the PM networks. So these findings combined suggest that to retrieve an integrated, detail-rich episodic memory, the AT and PM networks must interact with each other and with the hippocampus. So the final thing that we wanted to look at was whether or not there were any changes in network dynamics that were specific to whether people were remembering item information with a high degree of precision, or whether they were remembering spatiocontextual information with a high degree of precision. So we finally ran an analysis looking at network changes as scaled with the precision of their item color memory, which I'll show here on the left, as well as the precision of their memory for the scene perspective information, which I'll show you on the right. So because we're looking at this remember period, we're going to be looking for network changes that are unfolding in parallel and are differently related to how well people could remember the item color or the scene location. So what we ended up finding was that object memory precision was associated with an increase in functional connectivity between the perirhinal cortex and amygdala, two key nodes of the AT network, with key nodes of the posterior medial network, including the precuneus and the angular gyrus, as well as with the hippocampus. Now, in contrast, scene memory precision was associated with an increase in functional connectivity from the parahippocampal cortex and retrosplenial cortex, which we know are involved in scene processing, to these same medial and lateral parietal nodes. So these results suggest that the contents of memory, so whether people are calling to mind item information or spatial information with high precision, biases the observed pattern of corticohippocampal connections. And moreover, memory precision for both the item color and scene precision were associated with changes in network connections that converged on the angular gyrus and the precuneus, which are two areas that have been proposed to play more domain general roles in episodic recollection. So the lateral and medial parietal cortex then seem to be an important point of convergence as memory details are reactivated through the hippocampus and then integrated into memory. Now, Rose and I have talked about this in the context of a broader model of how regions in the PM network work together to support episodic memory and other forms of construction. And I have an overview of this framework shown here, where these parietal areas are interacting with content-specific areas, for instance, those in the ventral visual stream, to integrate, image, and refine the contents of recollection. So in other words, they take a bunch of reactivated bits of information, stitch them together, and then make them feel like a memory. And very likely a similar type of process is going to unfold for any type of mental or episodic stimulation. So moving forward, we're interested in further interrogating the subnetwork organization of the PM network to figure out how information is flowing through this network to support different aspects of event perception and memory. So I'm going to wrap up there, and I just want to thank the members of my lab, especially Rose Cooper, who ran that fantastic, well, she did a fantastic job running the last study that I showed you, as well as my collaborators on the earlier work that I showed you, Charan Ranganath, Andy Olina, Xiaofang Wang, and Laura Libby. And thank you for your attention.
I'll do a real live clap instead of a Zoom clap. That was beautiful work, Maureen. Thank you so much for coming and sharing it with the group. If anyone has questions, please just chat them out, write them out in the chat box if you'd like. I'm sure that Maureen would be more than happy to have a chat with you before we switch over to Xenia's work.
Hi, Maureen. This is Rosanna Olson. I have a quick question.
Yeah, hi.
Thanks for the beautiful talk, as always. I just wanted to ask you a quick question about the Wang 2016 study that looked at anterior perirhinal versus posterior perirhinal. And perirhinal cortex, as you know, is a humongous area, especially the way I define it in my lab. And I was just curious, you know, some people subdivide the perirhinal cortex into Brodmann area 35 and 36. And I was curious if the former RA looked at whether the alignment of those functional regions, whether that aligned to any known structural parcellations, like is it more 36 versus 35, et cetera?
Yeah, I think the most anterior portion better aligns with area 36. Now, in this analysis, because we took the somewhat coarse approach of just slicing up along the long axis, so then we're losing anatomical boundaries within each one of those slices along the long axis. So I can't tell you anything about any more fine-grained boundaries than that. But that was the literature that we were trying to relate those results to, is that there does seem to be some sort of division there.
Thanks.
So I see there was a question in the chat about whether any of the findings were different for the left and the right sides of the brain. I think more or less, we don't see strong hemispheric differences. I believe, if I am remembering correctly, and I don't think, unless Rose is here in the chat and can correct me, but if I'm remembering correctly, in the last study that I showed you, we did limit the analysis to left-sided ROIs, and that's because, at least in the context of these parietal regions that are involved in memory retrieval, the left angular gyrus tends to show up more consistently than the right angular gyrus. And we wanted to simplify our analysis approach by focusing on one hemisphere at a time. But I think more or less when you look at the other hemisphere, things don't look qualitatively different, although they might look quantitatively different.
Okay, thank you.
It looks like there's another question asking about how the border between anterior and posterior PRC was defined.
Yeah, so here it was totally a data-driven analysis. So we didn't impose any type of specific guideline between that anterior and posterior PRC. So let me see if I can find that slide real quick. Yeah, so this division here was entirely just based on the similarity of these slices in our connectivity with the rest of the brain. And then she used an analysis approach similar to the community detection, although it's a slightly different procedure, to identify which slices tended to lump together. So this was not based on any anatomical marker per se. It was purely just based on their functional connectivity with the rest of the brain.
Awesome. Are there any lingering questions that anyone wants to throw out there before we transition over? If not, no, we have a thank you. If not, I will reiterate that and thank you, Maureen, as Ramazana said, for a beautiful talk and for taking the time to be here with us today. It was great.
Yeah, thank you for having me. I was excited about speaking to this group in particular because I'm continuously in awe of how much you all know about the hippocampus and the anatomical specificities of the hippocampus, whereas I tend to focus more large scale on these cortical networks. So I'm curious to hear more about Xenia's work and your thoughts.
Awesome. All right, Ravan, I will switch over to you.
Yeah. So I'm very happy to introduce our last speaker for this 2021 series with Xenia Grande. So Xenia is a PhD student at the German Center for Neurodegenerative Diseases in Magdeburg, Germany. Her research is focused on the functional architecture within the medial temporal lobe, and she is most interested in how high-level cognitive functions like memory emerge from the interactions of subregion within the human parahippocampal system. And today she will be sharing her work, information flow within the human entorhinal hippocampal circuitry. So Xenia, the floor is yours.
Yeah, hello everyone. I hope you can see my screen. So thanks a lot for the introduction, and I'm very grateful for the opportunity to talk today to you about the information flow within the human entorhinal hippocampal circuitry. So thanks a lot to the hippocampal subfields group for that. Let me start, if it works. Let me start like Maureen already introduced to us the large-scale cortical systems that are presumably processing item and context information. So these anterior temporal and posterior medial systems, they enter the medial temporal lobe, as Maureen already said, via the perivinal cortex and the parahippocampal cortex. So now we would like to ask how they actually influence the information flow in the entorhinal hippocampal circuitry. So why are we interested in that question? First, this circuitry is essential for the formation of multimodal representations that serve memory-guided behavior, but also knowing the information flow in the circuitry may inspire neurobiological theories on the cognitive impact and progression of Alzheimer's disease. So with that in mind, let's turn now to the anatomical structure of the entorhinal hippocampus circuitry. As you may have noticed, I talk about the human brain mostly, but for all of those who are more convenient with rodents, there is a quick reference here. So in the human brain, the entorhinal cortex has roughly such a shape. And if we go posterior from there, we find the hippocampal body and also the subiculum and subfield CA1. And if we want to describe the positions in the entorhinal cortex in the transversal axis, we can use the terms medial to lateral. And in the subiculum and CA1, we use the terms distal to proximal. And this just refers to the distance from the dentate gyrus. So these regions come with a certain wiring pattern. And roughly spoken, there are parallel connections that split the entorhinal cortex CA1 and subiculum regions in their lateral, medial, and distal proximal parts, because those are respectively connected to the perirhinal and parahippocampal cortex. Note, however, that also profound crosstalk exists here. So this wiring pattern actually influenced proposals on how information flows here. And as we learned already from Maureen, the perirhinal and parahippocampal cortex, they receive input from the cortical systems, presumably processing item and context information. So it's thought that also along these lines, the information flows, maybe even into the hippocampus or within the hippocampus. So when I project this idea back onto the 3D view, we end up with this stripe-like pattern, let's say. So that's our rough proposal. Empirical evidence for this exists in many studies that examine functional connectivity profiles and also differences in information processing within the subregions. And without going into any details, I just want you to see that the evidence is very much scattered across different studies, methods, species, and also has many nuances regarding the exact divisions. So this all motivated us for the current project, as we aim to identify, or better to unify the advanced previous research by asking two questions. Via which routes do the human entorhinal cortex and hippocampus subregions communicate, and which information is processed where? So therefore, we analyzed the functional connectivity and activity in the entorhinal, subiculum, and CA1 subregions and seek to find evidence for this schema here. We do so, or we did it in a single data set, and we subdivided the entorhinal cortex in 4 subregions to achieve a more fine-grained view. And this was inspired by recent work from Menno-Witter that showed considerable high complexity of the primate entorhinal cortex. So to do all this, we used 7 Tesla imaging and acquired functional data during which participants performed an item context memory task. And with that, we just dive into the first question: via which routes do entorhinal cortex and hippocampal subregions communicate with each other? So basically here we want to identify functional connectivity profiles that are completely independent of the task at hand. And we did that in a seed to voxel-based manner, where we take seeds in the entorhinal cortex. So what we wanted to use are functionally relevant subdivisions of the entorhinal cortex. That means they actually should transfer some signal further from the anterior temporal and posterior medial cortical systems. So therefore, we did a short pre-analysis, or not short, but pre-analysis, and we correlated the activity in the entorhinal cortex with the endpoints of the 2 cortical systems, the perirhinal cortex and the parahippocampal cortices. And this basically gave us medial and lateral regions in the entorhinal cortex that we could divide again into an anterior and posterior part. So that's our 4 subdivisions. And now we were curious to see via which routes these functionally relevant subdivisions of the entorhinal cortex communicate with the subiculum and CA1. And we identified connectivity preferences for each and every part here. And we see that the subiculum CA1 border primarily connects with the anterolateral entorhinal cortex, but that the distal subiculum and also a bit the proximal CA1 rather connects to the medial entorhinal cortices. So if we put this together now, we see that information in the entorhinal hippocampus circuitry may travel along these routes. But it doesn't tell us anything yet about the type of information that's processed there. So that's the second question. And for that, we examined from all the entorhinal and hippocampus subregions whether they rather process item or context information. And herefore, we could take advantage of the task that the participants performed, as they always had mini blocks in which they processed either item or context information. So let's first look into the entorhinal subdivisions. From each individual, we extracted parameter estimates for either item or context processing. And I depict here the parameter estimates for the item condition in red dots and lines, and for context in blue dots and lines. And the lines are always the average across the individuals shown by the dots. So here in the lateral entorhinal subdivisions, these lines overlap, which shows that both subregions process item and context information similarly. In the medial subdivisions, however, this looks very different, as here the blue line is higher, thus they process more context information than item information. So if I just briefly sum this up for you, I put these icons here next to the entorhinal subdivisions as a symbol for the information. Let's look into the hippocampus and subiculum now. So for the subiculum, we did the same procedure. And what we find basically is that the distal subiculum processes more context information than item information. And we also similarly proceeded with the area CA1. And here we found that CA1 in general, and also the subiculum CA1 border, do not show a difference between item and context information processing. So again, to put this into the icons, it would look like that. Knowing all of this now, I would like to bring this together and look into the information flow within the human entorhinal hippocampus circuitry with you again. So based on our data, we think that context information is processed in the medial entorhinal parts, which posteriorly connect to the distal subiculum that also processes context information mostly. And in contrast to that, the lateral entorhinal parts, they process item and context information similarly and communicate anteriorly with the subiculum CA1 border that also shows similar item and context information processing tendencies. So with that, we replicate indeed earlier findings on the human entorhinal connectivity. And also we find indications that support recent findings on more of a multimodal processing in the lateral entorhinal cortex already. And moreover, we find actually less segregation in the CA1 than it was suggested by rodent work. So very briefly, why is this all interesting now? Basically, with that, now we can see how our findings may have functional implications. So we find areas, as you may have noticed, that process specific context information, but not areas that process specific item information. So we wonder actually whether this may relate to the idea that there's a contextual scaffold that is provided in the hippocampus to support the formation of memories. And second, there is some clinical implication probably, as early tau pathology progresses along the anterolateral entorhinal cortex connectivity pattern that we also see. So this may lead us to think about associated cognitive impact. So overall, I think our data shows that it's not only subregions, but also even the within subregional level that may hold very important insights into function, and that there is a great potential here for understanding how cognitive function emerges from the interaction of brain regions. And with that, I would like to thank especially the people without whom this all wouldn't have been possible, mainly David Barron for his wonderful supervision, and also Magdalena Sauvage and the members from the Duzel lab for their support, and of course, Emra Duzel himself. And thanks again for the Hippocampus Subfields group for the initiative, and you for your attention. And if you have any questions, you can always write me an email. Thank you.
Wonderful. Thank you so much, Xenia. It's amazing to see the progression of what you can do with 7T data. Like that's just a general statement of wow. And I think there were such complementary talks that you and Maureen gave in terms of going broader and then a little bit, a lot more specific. So I will say, yes, you can send her a message, but if anyone would like to ask questions now, I think that would be great. There was an easy question for you. Are these data published?
No. That's an easy answer. Or let's say not yet.
So there's two questions. The first is, have you tried data-driven methods like ICA instead of seed-based approaches?
We didn't try it. The reason for that was mainly that we wanted to be very comparable with previous research that Anne Maas and David Barron did for the entorhinal connectivity profiles. Yeah, so that's why we just follow their approach, basically.
Awesome. And then Noah often has a question about if you could expand more on the task that was used.
Yes, I can certainly do that. Let's go back here. First, keep in mind that we actually did not really take the task here, but we only took advantage of the fact that the participants were processing items or objects or scenes. But doing the tasks, they were actually, it was kind of a pattern separation task. So they were instructed to focus on the images and to see whether they changed actually. It's like a two-back task for that. But we did not differentiate according to encoding versus retrieval moments or stuff like that.
Great, thanks. All right. Two compliments, brilliant and great. And there's a question from Ty Wisher. Do the number of voxels within ERC seeds play into the functional connectivity results?
Yeah, that's a very interesting, good question. We did not explicitly check for this. We just made sure that basically the just for left and right hemisphere, the amount of voxels would be the same, but we really wanted to use the functionally relevant subdivisions. So we did not really check yet for the size of them. And maybe to add, this may not explain actually the results that we find this kind of stripe-like pattern as well. And yeah. So.
Awesome. Thank you. I think we have another compliment. We have time for one more question if anyone wants to ask it. I don't want to cut anyone's interest off. Oh, there's Rosanna. There she is with her.
You can always count on me.
I know you have your exception to the rule.
Great talk, so fascinating. I was just curious, David and I have talked about this issue about, medial and lateral ERC and what anatomical connections they have with other regions. And he had mentioned to me private communication that maybe you've been involved in these analyses too, about looking at connections with retrosplenial cortex. And I was just curious if you've looked at any of that here and how that relates to the circuitry that you described.
Yeah, that's certainly something we have in mind, especially given Manovich's work. We did not specifically look into that yet. I just saw basically yesterday, I ran an analysis on that. I saw basically just yesterday that the subdivisions we have right now, they do not seem to be the ones that are mainly connected to the retrosplenial cortex. So in the entorhinal cortex. So this might be another part actually of the entorhinal cortex that plays a role here.
Fascinating, thank you.
Awesome. Well, thank you for sharing that. It was also a very beautiful talk. And I think both of you did such a great job sharing your work. And again, thank you very much on behalf of the entire Hippocampal Subfields group for joining us, to our speakers, but also to our audience. The enthusiasm and the participation is really encouraging. And I think it speaks volumes to the community that we're building. And I can't wait till we can have more communications in as the working groups pick up, and also in having these webinars in 2022. A few quick notes before we go. As Anna mentioned, we will be sending out a survey. We invite speakers, but we also want to know people who are interested and have work that is coming out and that you'd like to share. So we'll ask twofold. What did you like? Maybe what could we do better? What topics do you want to see? And also, are you interested in sharing your work? So keep an eye on your inbox. That'll be coming to you next week. And a reminder that this will be posted on YouTube, so that if any of you want to go back, I know that I like to go back to the talks when I'm less distracted by my stress about tech issues, to make sure that you are checking us out on YouTube, especially if you've missed any of the webinars that you might want to go back to. So with that, I will just say thank you. And Raban, do you want to add anything before we sign off on this set of talks?
No, not really.
Okay.
It was a pleasure to build this webinar series with you, Kelsey, and I would like to thank everyone who participated.
Yeah, it's been nice to work on this with you too. So thank you to everyone. Good feels all around. And yes, as Rosanna said, please give us feedback. We want to keep doing this, and we hope to see you before the webinar series. Hopefully the world opens up to where we can have a meeting that we can all grab coffee at or something at soon. So with that, have a great summer, everyone. And thanks again.